Thank you very much. So I'm really feeling special today because well, D-IAS was just officially opened last week, and so I was invited to give this first seminar speak today. So I thought that maybe before I start talking about some scientific things, I very briefly add a little bit of information to well, my background and what we are actually doing here at SDU in well, just general words. So that's why let me start with this slide. So this introductory slide to my group here, and so we call ourselves a computational biology and computational physics group. And well, this is a photo more or less of all the people that are associated with the group right now. And I personally come from physics, actually. So I got my PhD in physics, and so I did it in Germany, in actually Frankfurt Institute for Advanced Studies, which has some parallels with the D-IAS here, as you can imagine. And well, right now, the main focus of our research challenges are outlined here. So we are very interested in looking at various problems in biology from a physics point of view. So we try to understand complicated biological phenomena by using well, approach as a physicist would choose. And also we do it by employing a so-called computational microscope, as this is basically the focus of my talk today. And so we also do some method developments and well, basically develop new techniques that permit us studying more and more complicated problems related to biology. So here is just sort of well, a sketch of the different scales of systems that we are in principle interested in. I should warn you that I'm probably not going to talk much about birds today, or maybe well, cells and organs, but I will spend some time talking about molecules. Well, I was asked to give actually a very brief presentation, so just limited by 20 minutes. So that's why, unfortunately, I cannot talk about all the different things that we are doing here at SDU. But I hope there will be some discussion following my talk, and then we can, in principle, discuss more things if you're interested in. All right. So I would like to start with actually going a little bit back in history and reminding you basically the way how different communities of scientists well, approached different questions. And so in physics, for example, there are always computational, theoretical scientists, and there are also experimentalists. And for a very long time, so these two groups of people were actually, I wouldn't say they were fighting against each other, but they all often had a feeling that they're actually doing more important research than the other group. So for example, if you go and talk to a hardcore experimental physicist, well, in previous years, so you often could hear an opinion that, well, you know, we do experiment, and this is how the life is. And I mean, well, the computations and theories, in the best case scenario, can just support our observations. And you could also actually hear quite a different opinion if you talk to theoretical people. And I put this quote here on purpose because I wanted to demonstrate that actually in the year of '98, there was actually a Nobel Prize awarded basically for computational theoretical research in chemistry. And so basically with this, I would like to start by kind of showing you that this basically acknowledged that computational and also theoretical science is actually an important area, and that actually provides new insights into, well, how things are in nature. Well, but well, this statement is sort of a little bit weird. I don't fully agree with that because I'm pretty sure that well, most of you also would say that, well, definitely well, there are a lot of experimental well, questions that cannot be addressed by just well, putting everything inside of the computer. But today, the point of my presentation is actually to show you that some questions can really be solved nicely and accurately and precisely using modern computational techniques. Well, but also I think that, well, most of you should probably agree with me that so there are certain fundamental laws of nature. And so those laws of nature are obeyed always. So, well, Newton equation, Schrödinger equation, I mean, well, we don't really argue that, well, these equations are really the fundamental laws, and, well, you know, if an apple falls down from a tree, well, this is how we are going to describe its motion. Or even if you shine light on in this double slit experiment, well, you're going to see some interference pattern, and, well, I mean, this is basically the way how you can describe what is happening in this case. So in a way, so one could basically state that so the world around us is actually governed by equations. So and every phenomena, everything that we see and observe, it can actually be understood if we know the right equations. And well, those are just really the basic fundamental examples of what one has to use if you want to describe this school examples. So also would like to say that actually most of the phenomena is actually driven by not only by equations, of course. I mean, equations is basically what we take from mathematics, but one also has to understand some physics behind things that happen in everyday life. And physics-driven examples basically allow us to construct computational models, and also in other words, to make some simulations on a computer that actually would not be possible to do otherwise. So it wouldn't be possible, for example, to take a microscope and observe how these atoms in a complicated biomolecule would move. So I mean, there is no way how you can have a microscope that would have a resolution like that. But using computer, we can do that. And this is not just some fancy computer game, because here we are really trying to take the right equations that describe the motion of atoms, in this case, in this biological molecule, and observe how they move. Well, I'm not arguing that if it is important or not to know how this particular atoms move, but this is just an illustration to show you that, well, I mean, you can do it using computer. Another more complicated example is shown here. So here you see a DNA double helix that, is supposed to propagate through a hole in a synthetic nanopore. So this has actually been realized as an important example in nanotechnology, that basically people suggested to use synthetic nanopores for DNA sequencing. And there was a lot of computational efforts in this area, basically that demonstrated what kind of nanopores you should construct in order to get the right device. Well, there are actually a lot of different examples where basically I could be talking for hours about basically most of this highlights that are shown here. And so all of these examples and all of these exemplary systems, they actually can be described computationally if we know the right laws that describe the motion or certain of atoms or certain phenomena. Like, for example, so in this case, so this is an illustration of an ultra-relativistic particle that propagates through a crystal. And so you can, for example, try to model how would this ultra-relativistic particle propagate, and what would be the trajectory, and what would be the the associated effects that arise when it goes. So in this case, you need to solve the relativistic equations of motion, and so this can be done if you have specialized software that allow you to solve these complicated equations computationally. Well, but since I'm limited in time, and so I also want to be a little bit more specific today, so I will be talking mostly about biomolecules. And this also, as the title of my talk suggests, so I will be talking about some biophysical applications. So that's why, I mean, this is the niche of that I will be looking at. And so those other examples is just to illustrate you that, so in principle, nowadays, so we can really address and model really complicated systems and processes. And so in this case, so all of these systems were actually studied with a software called Ambient Explorer. So that is partially developed in my group here at SDU. All right. Well, but now, so maybe some of you were curious, what is actually this computational microscope I put in the title? So, and I think that the best way how I can introduce it is actually by this slide that so I actually inherited from my colleague Klaus Schulten, with whom I worked in the USA before coming here. And so basically, the computational microscope is not really a microscope in a way that we are used to. So it's not something that we have to fine-tune and then look in some glass and then see something magnified. So it's actually a set of approaches and a set of tools that allow us to see and model the most peculiar effects in a computer. So, and here, I mean, these are basically the 5 different components that this computational microscope should have. First of all, since we are interested in biomolecular systems, it has to be dictated by chemistry. I mean, you have really to know what systems you are looking at, and you need to know how are the atoms bound together. So what are the different bonds between atoms? So are you studying proteins, DNAs, I don't know, membranes? So, well, this has to be known. And I mean, this is basically governed by chemistry, and well, if you're a scientist working in a certain area, well, you're supposed to know what you're working with. I mean, otherwise there is no real point in doing, well, step 2. Then physics is the second component. So as I was saying before, well, physics is something that is really basic in many of these examples that I mentioned in the previous slide. And well, in this particular case, so often so the Newton equation allows to a fairly good approximation to model certain phenomena in the computer. Of course, it's not always the case. So sometimes you have to use other approaches and other approximations. Well, in this example, in this slide, just an example. Well, we can't really live without mathematics because, okay, if we have the right chemistry, we have the right physics model set up, we still need a way how we can actually solve the complicated equations for the system of our interest. And in this case, well, you need to find out some mathematical tricks how you can do it efficiently. And well, be aware that, of course, if you're dealing with biomolecular systems and we would like to study how all the individual atoms move in these biomolecular systems, we cannot really do much on just a piece of paper with a pen. I mean, so those times are kind of over, and well, I mean, they never started for these big systems actually. So we need some software. So we need a program that actually would allow us to solve the mathematical equations. And so in this case, so this NumD software is just one of the examples that can be used in order to solve the right equations of motion. Important here is that this software is actually tuned and coded in a way that it can be used on so-called supercomputers. So supercomputers is a bit different than just a normal workstation that I hope all of you are familiar with. It's basically a set of many computers connected together with a highly efficient, fast network. And this way, so you can actually solve so these equations very, very fast and very many times. For example, if you would like to study molecular dynamics, so see how the atoms move, so you probably need to solve this equation like, well, 1 billion times. Yeah, and I mean, of course, if you don't have a powerful enough computer, well, it's quite challenging to do so. All right, so I also thought it may be a good idea to tell you maybe a few things. Why is it actually worth studying biological systems inside of a computer? I mean, you could tell me, well, I mean, biological systems, well, they're already big enough, so we can just go in the laboratory and then take the normal microscope and then basically learn everything we would like to know. This is true to some extent, but here I outlined some points that I believe are important and can be efficiently attacked by using computations. So, for example, in this graph, so you see that this is the tendency of how molecular dynamics, all atom molecular dynamics, was evolving in time. And so here, on this axis, you see the scale of how many atoms it was possible to put inside of a computer. So now we are somewhere here, and so actually we are talking about systems that are on the scale of 1 million atoms. So you can really take a system of 1 million atoms, put it inside of a computer, and then see how all these atoms move coherently. And I mean, of course, if you're dealing with sizes like that, so we can really address a lot of questions that were not possible before. But also we can actually do it very, very efficiently, and we have the full control of the system when we have it in the computer. So in laboratory, often you cannot really control small things. So you just will have to invent certain ways out how you can, well, basically make the environmental impact on your studied systems less. And well, this is not the case in the computer. So in the computer, you can change whatever you desire. And this allows you really to look in the details of how these complex machineries work. So here, for example, so in 2014, there was a paper published where people studied the entire HIV virus capsid in all atom representation. And so this was actually for the first time somebody sort of suggested the structure of this HIV virus capsid. Of course, it has huge applications. If this structure is then confirmed in follow-up investigations, well, maybe we can design some drugs that would basically attack the weak points in this capsid, and well, maybe in the long run help solving one of the big issues in health science. All right. Another thing that I find is very important, and this is where the computers are very crucial, is that so if we look at the different scales involved in biology, so here I basically show you all the way from cell to individual molecules. So you see that, so here you can really nicely look at an entire cell in a microscope. I mean, there is no doubt about that. But I mean, there is no way how you can actually look at these small molecules inside of the cell. But now, the fun part is that actually the way how a cell works, it really relies on what is happening on the scale. Because if you change the interaction of this molecule just a little bit, by changing I mean that you don't change it by hand, but I mean you introduce, for example, some modification in the entire machinery, that it could have actually impact on the scale of the entire cell. So, for example, you could introduce a drug. A drug is typically a small molecule, so it would penetrate the cell, do some interactions, and then this would lead to killing the entire cell, or maybe vice versa, like prolong its life by orders of magnitude. So, I mean, this is what I call this, well, to make some analogies, a butterfly effect, basically by showing you that, okay, you have really small changes on the scale of atoms and molecules, and this has implication on the scale of an entire organism. Also, I should probably mention that, so the description that is required to model cells and molecules is actually changing from purely classical to completely quantum mechanical. I mean, if you talk about molecules, and if I would make a next step, I would be talking about electrons. I mean, I cannot model electron dynamics using classical physics. So we need to have quantum mechanics there. So in a way, so this bridge here shows you that actually quantum mechanics is really the fundamentally important component of how do the living organisms work. And so here I wanted to show you 3 examples that I find very, very interesting. And so actually all of these 3 examples are being investigated in my group. And those are just some of the examples that we're interested in. And these are exactly these examples where you see that something happens on the scale of an entire organism, and actually this effect has something to do with how the molecules interact. For example, this case here. So I don't know if all of you recognize, but this is actually one of the images I found online of skin cancer. And this could happen if you expose the skin to UV radiation for a sufficiently long time, then it can actually get damaged. And I mean, in the more extreme case, it can even lead to some things like cancer. But the interesting thing is that in order to understand how it develops, we need to go all the way to the scale of molecules. And well, here you see this DNA molecule. And actually, what happens is that this DNA molecule gets damaged by UV radiation, and then so basically this has a consequence in a multi-step process that leads to developing of these cancer cells. So now, in order to understand how to cure that, we actually need to understand how to model processes on the scale of molecules, how to model the repair of this damaged DNA. Another example is aging. So I mean, well, this is another illustration I found somewhere. I find it very nice that you see that, well, here this young girl develops in an old lady, and this is basically happening with everybody of us. And the question is, why does it happen? What is actually going wrong? So why do people live well the years they live? And one of the reasons that was discussed largely is that this has to do with formation of free radicals inside the cells. And these free radicals, they just emerge spontaneously as side reactions of some normal functioning of biomolecules. And so here I show you another example, basically of a molecular system where such a radical could be formed. And again, you see, we are talking about scale of molecules. So now, in order for us to understand how these free radicals are formed, we need to go all the way to the scale of molecules, and then maybe we can say something about formation of these radicals, and then learn what impacts they have on the scale of an entire organism. Well, the third example here is also very interesting, and to be honest, I learned about that about 4 or 5 years ago, and I was actually surprised. So it never happened to me before that I didn't realize that apparently this sense of smell is something that is also not completely understood. I mean, I always thought, okay, well, there are these 5 basic senses, and we all know exactly how they work, and the question is closed. But apparently, this is not the case. So apparently, there is really a huge debate about how how do people and animals sense different odorants, and how they can distinguish between different scents. And talking about that, so it brings us again to the scale, this very small scale that is not observable by the real microscope. So we really need a microscope that would allow us to look at all the small details. Well, and if you're still not convinced that a computational microscope is something that is important and useful, so there was another Nobel Prize awarded in 2013, which was awarded, again in chemistry, for the development of multi-scale models for complex chemical systems. And I mean, this is not, of course, exactly for this computational microscope, but you see the parallels. You see that, so these people, they were awarded the Nobel Prize for developing methods that allow to study complicated biological systems. All right, well, now, in order to start using this computational approach, so we need to choose the right equations to model the systems and phenomena that we are interested in. And as I was telling you before, I mean, there are many different types of equations, approximations, models that we can put forward. Important is always to realize, if you make a model approximation, so what is the limit of this model, and so where should you stop, and where can this model be applied? So here, basically, in order to study biological systems, we need to combine classical molecular dynamics with some quantum physics and quantum chemistry. So this will allow us to study the dynamics of heavy atoms, while this method will allow us to study electronic dynamics. And well, I was talking about that already. So here we are really nowadays are capable of studying really huge systems like HIV virus capsids, photosynthetic complexes, and so on. So here, well, there is still some space for development, and so unfortunately, this is basically the limiting scale of what we can do nowadays. Of course, it depends on the methods, and I mean, my quantum chemistry colleagues could probably argue with me about that. So also, I should say that we are actually quite lucky that actually we do have a special computer called Abacus 2.0 here at SDU. And so this supercomputer can be used efficiently to study the processes and systems I was talking about. And I mean, supercomputers are really becoming an important tool nowadays. So they are, implemented in many different countries, many different cities. And this is really like a must-have tool for computational researchers, because otherwise you cannot solve complex problems. All right, let me now give you a couple of examples of, well, this biophysical applications I wanted to talk about. And I basically wanted to show you in 3 separate slides. So these 3 examples I was talking about before. So this UV DNA damage, the case of aging, and then also the problem of olfaction. So here, I mean, this is this picture that basically shows that this DNA double helix at this particular site was damaged by UV radiation. So you see, it kind of looks a bit weird, right? So that something sticks out. And if this damage is not healed properly in time, this DNA will start working wrong, and then it will lead to some well, more complicated problems in the cell, which eventually could lead to formation of cancer. But luckily, there are special enzymes, special proteins that are designed by nature to cure this type of damage. So these proteins, they would find the damaged site, bind to it, and then fix it by doing some electron transfer. So here, this illustration, it's basically just an illustration to show you that we can use tools of quantum chemistry to model, transformations on this scale. So we can actually model how these electrons would move from this repair enzyme to the damaged DNA site. I mean, this is not happening in this video, I know, but it's just sort of a nice illustration to show you that, so we can really look at these details on the level of electrons. So another example related to aging I was talking about is when spontaneously, unpredictably, radicals are being formed inside of some biological systems. And so this, weird colored image you see here is actually a complex of proteins embedded in a membrane. And this complex of proteins is basically existing in every organism. It's really important for functioning of cells, because one of its important biological functions is to drive protons from one side of the cell to another. And I mean, this motion of protons is very important to accumulate enough energy in the cell to sustain in the long run. And this is happening through a series of electron and proton transfers. And when these transfers happen, sometimes it just happens wrong. And so then when it happens wrong, then there is a chance that free radicals are being formed. And these free radicals could lead also to killing cells and damaging important functioning of the living organisms. And here I wanted to show you this plot, which probably doesn't say you much immediately, but this is to illustrate that. So using a computational approach, so we can model how are the electrons being transported inside of this complicated machinery. So we can really look. So if the electron was initially located on one side of this protein complex, what should happen in order for this electron to go elsewhere? And well, believe me, I mean this is really something that computations can do. Well, I wouldn't say routinely, but it's feasible to do it using computer. But I don't really believe that looking at electron dynamics is possible nowadays with experimental techniques. So the third example I want to show you is about the sense of smell. And so here, basically, before I show you a video that I have prepared, let me just say a few words kind of to prepare you what is going to happen right now. So for a long time, it was believed that the sense of smell works in the following way, that molecules, volatile odorant molecules, they penetrate their olfactory receptors, and then depending on the shape of these molecules, it would activate the receptors. And I mean, this was basically the mainstream theory of olfaction, of the sense of smell. And then suddenly there was a discovery that apparently it's possible to distinguish between normal odorants and odorants which are deuterated. So deuterated odorants is basically, it's the same odorant where all the hydrogen atoms are replaced by deuterium. So it doesn't change the shape of the molecule at all. It just stays the same. The only thing that happens is that the hydrogens now become heavier. And when this substitution was made, so it was demonstrated that, well, fruit flies and also humans sense the smell differently. So here in this video, I mean, I basically wanted to show you one of the hypotheses that was suggested to explain this phenomena. So here you see a normal odorant and a deuterated one, and you see that hydrogens in the deuterated odorant, well, vibrate a little bit slower. I mean, this is because of the mass, that the mass is larger, so the period is larger. And then basically there was a suggestion made that these vibrations are really, really crucial. So these vibrations basically is what leads to distinguishing between the different odorants. And the way it happens is that an olfactory receptor is here seen as this bluish U-shaped structure. And it has probably an electron on one side of this receptor that desperately wants to get to another side. And this is not really possible because of some energetics inside of the receptor. But as soon as an odorant binds, it starts vibrating, and these vibrations, they assist the electron to flow from one side to another. And basically, so here we're talking about the interaction of an electron with some vibrations of the molecule. And I mean, this is a hypothesis which I believe is a very interesting one, and it has a lot of physics in it, but of course it hasn't been proven yet. And so here we are now looking at also at the details on the smaller scale to see if we can well, learn something for olfaction. So I skip this slide. So in the case if you would like to know about migratory birds and how this sense the Earth's magnetic field, I can always talk about that later. But since I'm a little bit limited in time, so I just want to get to the last part of my presentation, where I would like to show you basically what is the approach we use in the group to study all these different complicated problems. And well, as I was saying before, well, we need some kind of software. So we are using a lot of different software to model different kind of systems, different processes. And well, basically, I would probably be correct if I would say that we are talking about like, I don't know, 5, 10 different software packages that are really tailored for different purposes. And well, believe me, it's really, really complicated especially for well, younger researchers to start well, doing something, because well, you suddenly need to learn all these different technical things. You need to know, how do I set up a system inside of a computer? What software should I use? Well, if something doesn't work, why doesn't it work? And so on and so forth. And so this is why so we had this idea some years ago to develop a tool that we called Viking, and I will explain you just in a second why it is called Viking. That basically is one software to rule it all. So the idea of the software is that so you choose a problem that you would like to investigate, and then you have some really intuitive, I call it plug and play setup for the simulation that allows you to prepare your system for complicated computations on the computer. And then when you're done, so you can, well, use some fancy, high-tech, modern technology to visualize your results. So in this case, well, you're manipulating with this DNA double strand using a smartphone. So I mean, this is kind of the ideal goal, what we would like to have. Well, be aware we are not really there yet, but well, we're working on that hard. So Viking is actually a tool that, well, the abbreviation stands for Scandinavian Online Kit for Nanoscale Modeling. And so it's actually a web-based application that basically allows anybody who registers on the website to get access to using this computational software I was talking about. And the idea here is that through web-based application, so you don't need any special computer requirements. So you basically can access it from any platform, any computer, and then, well, prepare your system of interest for the simulations. And I'm really talking about simulations I showed you before, talking about this electron transport, dynamics of molecules, and so on. So, well, it's already existing in principle, but, well, I would probably correct by saying that, well, there are still a lot of things which are planned for the next years. And, well, here, this is just some snapshots of what is actually happening when you are setting up this simulation. So you're guided through a set of questions that basically you need to answer in order to prepare your system for simulations. Questions like, I mean, what is the structure of your protein of interest? So what is the temperature of your system that you are investigating? And I mean, maybe like what is the solution that you would like to look at, and so on and so forth. And so we are really trying to basically bridge this gap and make it accessible for people that have no experience with computations, but also that we hope that this tool could be used by advanced users that well, would still have the possibility to fine-tune and change all the small parameters if they have the intention to do so. Okay, I think that I came to the conclusion of my presentation now, and I was actually thinking quite some time last night, how should I conclude all that? So I hope you learned some new things and you got like a feeling of what we are doing in my group here at SDU. And I think that in the spirit of being curious and this D-IAS, I think this is probably one of the best slides I can end up my presentation by quoting this gentleman here who unfortunately passed away quite recently. And he was a Nobel Prize in chemistry awardee for femtochemistry. And I think that I completely agree with the saying that curiosity is really an important factor that drives a lot of exciting things. And so one shouldn't limit yourself to a particular problem, but really be open-minded and look at diverse problems across disciplines. And having said that, let me thank my group for all the hard work and great science and discussions. And thank you very much for your attention.